Welcome, Bio 440 General Microbiology Lab folks. This is Chapter 5 Microscopy, Part 2, Take 2. The previous videos were out of focus, so we're trying a different approach here. You'll recall in the last video we were going over the structures, the parts, and functions of the microscope, and we had just gotten to the stage where we were trying to place a microscope slide on the stage when we cut. Before continuing, I just wanted to go over a couple of concepts that will be important for us to understand the functions of some parts of the microscope. So the first concept that we'd mentioned before is resolution. And resolution, kind of my gut feeling for resolution is it's the clarity by which we can identify objects. And maybe the best way is to show a simple example here using our index card. So if we presume we have two small cells, two small objects that are placed very close together, and let's say that we are looking at these two cells placed very closely together, and we're using a microscope that has poor resolution, you can see in our little cartoon here that the cells, the outline appears blurry or fuzzy. It's hard for us to tell, is this a single cell, a single object that's kind of dumbbell shaped, or is it indeed two separate objects or two separate cells? And then on the right, we're looking at those two cells using a microscope that has good resolution, and we can see we have nice, clear, defined outlines of the cells. We can tell that, yes, indeed, these are two small cells that are really close together. So when working with our microscopes, we're interested not only in magnification but also resolution. So we'll go back here to our first card. And so when we are looking at bacteria, and bacteria are very small, average bacteria maybe 1 to 2 or 3 micrometers. Because they are so small, what we often do is we'll stain them to increase contrast, and then we'll look at our stained bacteria with our microscope. So we don't worry about contrast once we stain our bacteria. But what we are concerned about is resolution. So when we're working with our stained bacterial smears, we want to increase resolution. And to increase resolution, we want to increase the amount of light that strikes the specimen and then works its way through the lens system and strikes our retina. Each photon of light which strikes the retina will generate an action potential, basically pieces of data that then will travel back to the visual centers of the brain. And our brain will use those action potentials, those data points, to create the image of the object that we see. So the more light that strikes a specimen and eventually strikes the retina, the more data points our visual center has, and the clearer the image the brain can create for us. So to increase resolution, we want to increase the amount of light that strikes our specimen. And there's 3 ways we can do that. We can increase the variable light control, increase the rheostat. We can raise the substage condenser, which we'll show you here in just a bit. And the substage condenser will help focus light on our specimen. And the third thing we can do is open our iris diaphragm. You'll recall the iris diaphragm acts like the pupil of our eye, and by opening the iris diaphragm, we're going to permit more light to strike the specimen. Now, in contrast, when we're working with wet mounts, living microbes, most microbes have very little pigmentation. And that means if we're looking at a wet mount where they're alive, we haven't stained them, there's often little contrast between them and the surroundings. So to increase contrast, the difference between the microbe and the surroundings, what we want to do is increase contrast. And to increase contrast, we're going to do the opposite. We're going to make different changes to our microscope. To increase contrast, we actually want to reduce the amount of light striking our specimen. So we can decrease the rheostat, the variable light control, we can lower our substage condenser, and we can also close the iris diaphragm. And again, that's going to help increase contrast when we're looking at live, unstained microbes. We're continuing with part 2 of our microscope video. Just want to finish a couple more concepts. So we were speaking about how we're going to increase resolution using our microscope. And another consideration is, as we're increasing magnification, as we're working from our scanning lens, 4x lens, up to our low power 10x, high dry 40x, and then to our oil immersion lens, we need to remember that as the magnifying power of the objective lens increases, the diameter of the objective lens decreases. So I just made a little cartoon here, so we can pretend this is the diameter of the 4x objective lens compared to the diameter of the 100x objective lens. And I actually unscrewed the 4x, the scanning lens, from one of the microscopes, and you can see the diameter here, relatively wide diameter. And here you can just barely see the little tiny 100x oil immersion lens here, almost like a pinhead. And this is a problem because as the diameter decreases, that means the objective lens can harvest fewer photons of light. So that would cause a decrease in resolution. So to compensate, as we start looking at our specimen using the scanning lens, we'll have our rheostat, our variable light control, at a moderate setting, say maybe 5 out of 10. And then as we move up to higher and higher objective lenses, we're gradually going to increase the setting of our rheostat. So by the time we're working with an oil immersion lens, we will have our rheostat maybe on the highest settings, maybe settings 9 or 10. And again, that's going to help increase resolution. We're continuing our discussion of the microscope parts and functions. So what we're next going to do is slide our microscope slide into the slide clips. The slide clips are part of an apparatus called the mechanical stage, which will let us move our microscope slide precisely over the surface of the stage. So I'm going to take a break here just so I can get my slide fixed into the slide clips. So I've just slid my slide into slide clips, and we want to remember to release the slide clip gently. Otherwise, if you release it quickly, the clip could actually smack into the glass slide and break it. Now, this is a prepared slide of Spirogyra, a photosynthetic eukaryotic microbe, and it has a really thick cover slip on it. We can see it's a circular cover slip. Now, I want to make sure that I center our specimen, our microbe, over the hole in the stage where the light will pass through. And the way I'm going to do that is I'm going to move the stage using the mechanical stage adjustment knobs, which are below the stage. Let me see if I can get those in focus here. So here we have the mechanical stage adjustment knobs, and we can see that by moving the lower knob, it moves my slide from right to left, and by moving the upper knob, it moves the slide forwards and backwards. So again, I want to use those mechanical stage adjustment knobs to get my specimen centered over the hole in the stage. So let's see if I've accomplished that. Okay, looks like I've got it centered. That's good. All right. So since we're looking at a relatively large specimen, Spirogyra, it has pigment, it has chloroplasts, we could choose to lower the substage condenser, and beneath the stage there's a knob which controls elevation of the substage condenser. So you can see it's in the lowest elevation right now, so I could raise it, okay, or I can lower it. By lowering it, I'll decrease the amount of light striking my specimen. And then also part of the substage condenser, we'll see there's a little lever. Let's see. We'll see that there's a lever on the substage condenser. This is the iris diaphragm lever, and by moving it to and fro, we can open and close the iris diaphragm. So when we need more light, we'll open the iris diaphragm. When we want a little bit less light, we can close it. All right. And finally, we just want to recognize that on the microscope, on the base, we've got a light switch, our on-off light switch here. So I'll turn on our light. And on the opposite side of this particular model is the variable light control, the rheostat. And if the microscope has been put away in the cabinet properly, the rheostat should be on the lowest setting 1 here. When we start focusing on our microbe using the scanning lens, I'm going to suggest that we start at a moderate level of light, say setting 4 or 5, and then as we increase magnification, as we move to low power, high dry power, and ultimately to oil immersion lens, we will increase the light intensity until when we're working with the oil immersion lens, we'll be working on the highest setting, say 9 or 10. Okay, right now I'm going to bring this back down to about setting 5. Towards the bottom of the arm of the microscope, we can see two focus adjustment knobs. The outer one is called coarse focus adjustment, and you'll see if I rotate it, there's relatively large changes in the elevation of our stage. And then there's an inner knob, the fine focus adjustment knob, and we can see as I rotate this, there's very small changes in the elevation of the stage. Now we have really important rules with regard to which focus adjustment knobs we use with which objectives. So our rules are, we only use coarse focus adjustment with the least powerful to shortest objective lenses. So we use coarse focus only with our 4x scanning lens and our 10x low power lens, because the more powerful high dry 40x lens and oil immersion lens, since they're so long, we're almost working with the lens almost touching our glass microscope slide. So as a consequence, we don't want to risk damaging the objective lens or our microbial specimen by ramming the stage and slide into the end of those objective lenses. So our rules are, when we're using the high dry lens and the oil immersion lens, we only use fine focus. Now, our scopes are lovely in that they are described as parfocal. That means once we have our specimen in focus using one objective lens, say for example the scanning lens, as a parfocal scope I should be able to change objectives and the specimen should remain in focus. Now our scopes are not perfectly parfocal. So what we'll be doing is we'll be getting our specimen in focus. We always start out with scanning lens. We'll be using coarse focus. Right? And we'll just describe how we're going to have our microbiology folks focus a specimen. So when we place our slide on the stage, we're going to look from the side. We're going to start with our scanning lens in place, and using coarse focus, we're going to raise the stage all the way up. And we're watching from the side to make sure by accident the stage and slide don't ram into the objective lens. It's only at this point that we're going to then look through our oculars, our ocular lenses, and then we're going to bring our microbe in a specimen by lowering the stage, lowering the slide. And the reason we do this is for safety's sake. If we focus by lowering the stage, we'll never have an accident of ramming the stage and slide into the objective lens. Okay? So I'd be looking through my oculars. I'm going to bring my microbe into focus using coarse focus adjustment. Then I'll revolve the turret or revolving nose piece or rotating nose piece. That click is important because it's telling me now that my low power lens is in alignment with the lens and mirror systems above. And now in theory again, the microbe should be in focus, but I might need minor adjustment of focus, so I could use maybe a minor adjustment using coarse focus or minor adjustment using fine focus. Now once the specimen is in focus there, we're going to rotate, again looking from the side, we're going to rotate and bring our high dry, our 40x lens into place. Now the question is, which focus adjustment knob can we use with a high dry lens? And you'll recall we can only use the fine focus adjustment, this innermost one, and it should only take half a turn, one, maximum 2 turns to bring your specimen into focus. If it takes more than 2 turns of fine focus to get the specimen into focus using high dry or oil immersion lens, make sure to call one of the instructors over, one of our great micro tutors, because it's possible something's wrong with the microscope.